Hi everyone, welcome back to the next review video for the biology or living environments regions. This is Unit 2, Topic 3, or enzymes. So, first, under enzymes, we have what are enzymes? So, enzymes are proteins. These are one of the key things that proteins are needed for, and enzymes are really necessary for all of our survival. So, what they actually are biological catalysts. And what that means is that their role is to speed up all chemical reactions without being used up. So their role is to perform various chemical reactions in the body, including digestion or synthesis, or hydrolysis or dehydration synthesis, as we learned about them earlier. And their key thing is that they do not get used up, so you can continue to use them over and over again. The last important thing you need to know about what an enzyme is the lock and key model. So enzymes are like the lock, which have their fixed shape, and then the key are like the substrates that bind to them. They fit its specific, and they have a specific shape to perform their specific function, and only their specific substrate combined with their enzyme. So it's like only one key. This key will only fit this lock. If you try to put this key in a different lock, it obviously won't go in and it won't unlock it. So you need to have a specific substrate for the specific enzyme. So now we have the components of enzymes and the substrates that bind to them. So first we have what is a substrate, also called a reactant. So the substrate is what the enzyme binds with at the start of the reaction. So the reactant is really, or substrate is really what you start with before the enzyme does anything. Next, we have the active site. So the active site is a part of the enzyme, and it's the location where the substrate binds. So the active site is on the enzyme. It is a specific portion which the substrate will then bind to, and this is where the lock and key model comes in. So the substrate shape is specifically matched to an enzyme's active site. Once the enzyme and substrate have bound together, then you get what's called the enzyme-substrate complex, which is just the combination of the enzyme and the substrate. So this is just the specific term for when the substrate is attached to the enzyme and it is undergoing whatever chemical process it's doing, whether it's digestion or synthesis. And then the final thing we get is the product, which is exactly what it sounds like, and that is what is created. So the product, you have the substrate, it comes in, it binds to the active site to form the enzyme-substrate complex, and then the enzyme spits out the product. So it could either be two substrates that are put together into one larger substrate, or one larger substrate digested into two or more various products. So down here we have an image of that process. So we get first this substrate. This is the enzyme's active site right here. This specific shape that fits the enzyme's shape the substrate shape right here. The substrate will then bind into that active site to form the enzyme-substrate complex, where the enzyme and substrate are connected at the active site. And then finally, you get the two different products. So this is a digestion reaction, as one large substrate is split into two different products. And the other key thing to notice is that the enzyme right here is the exact same as the enzyme was right here. So the enzyme does not get changed, which means that it can be used over and over again. So this reaction can occur multiple times with the exact same enzyme, which is a key thing to remember. The enzyme doesn't change, only the products do. And now, the final thing you want to know about enzymes are that they have specific optimal conditions. And these vary per enzyme, as we'll see in a minute. But you want to know that enzymes denature, and what denaturing is another word for losing shape and no longer functioning, because as we know, structure determines function or shape determines functions. So enzymes will denature and lose their shape if temperature is past the optimum. So on the right here, we have an example graph. They might ask you to draw a graph like this. So what you want to know about these graphs is that as temperature increases towards the optimal, the enzyme activity will also steadily increase. So the closer you get to the optimal, it increases. And then the second you get past it, there's a very sharp decline. Because if it gets too hot, the bonds that form together the enzyme and the protein will start to break apart, and the enzyme then denatures, which we see here with a rapid decrease in enzyme activity. So the closer you are to the optimum temperature, the better it is. And once you get past that, or be higher than that, there's a rapid loss in enzyme function. And then the second one you want to know is that there are also optimal conditions for pH. So enzymes denature if pH is too high or too low. So this is one thing that's slightly different than temperature. While if temperature gets really low, enzyme activity is also declined. It falls off or declines a lot faster when it gets too hot with temperature, whereas with pH, if pH is too low or too high, it will have an equal effect. So if pH is too high or too low, enzymes will denature. And we can see that over here with the graph of trypsin and pepsin, which are two different enzymes. We can see right here at their peak, it would be their optimal. So for trypsin, it would be about like around 8, and for pepsin, it's around here. So it would be about 3, pH of 3. And what happens when you get too high is that the function of each enzyme begins to steadily decline. And what might be good for one enzyme might be different for others, because all enzymes are all across the body, found in all various different conditions. Like some are in extremely acidic environments, like in your stomach, whereas others are in more alkaline environments. So one pH, like a pH of 8, is perfect for trypsin to function, but pepsin would be absolutely destroyed in 8. It has zero relative activity. So it's all dependent on the specific enzyme. Okay, so now we have some review questions. So the first review question we have is number 35. A cell with receptors for 2 different hormones is represented below. And then we have the picture of the cell here. Which chemical would most likely interfere with the activities of hormone A, but not hormone B? So this is a question really about the lock and key model. So we see hormone A is circular, so it would likely bind to these circular receptors here. And hormone B is square, so it would likely bind to these little rectangular ones over here. Whoa, don't know what that is. So we want to see a chemical that would interfere with A but not B. Because we know that chemicals only bind with their specific shape, we want to see one that would fit the shape of A. So we're looking for which one is a circle. So the answer would obviously be number 1, because that's the only circle one. And this would interfere with A because this could easily bind into these receptors, but it would not fit into hormone B's receptors, the rectangular ones. So it would interfere with A, but not hormone B. Okay, our next question. Number 25. Students did an experiment comparing the activities of 4 different enzymes, A, B, C, and D. The results are represented in the graph below, and we see a graph of pH and relative rate of enzyme activity. A valid conclusion based on the information in the graph is, and then first is that the pH of some enzymes change as the temperature changes. So this one would likely not be our answer because we don't see anything about temperature anywhere in the graph. It's just kind of throwing that in randomly, which isn't our answer. Next is that enzymes change color in proportion to the rate of activity. Again, it's about pH, which has nothing to do with color, so number 2 would also not be our answer. Next, a difference in the pH of an environment changes enzyme activity. So this one would definitely be our answer because we can see that as pH changes, the rate of enzyme activity also changes, which means that pH has a direct effect on enzyme activity. And just to be safe, we'll look at number 4. Enzyme activity causes acids to change into bases over time. This is just not true. We're changing the pH, not saying enzyme activity changes the pH of other substances. So number 4 is also a misreading of the graph. This one is more about, you can get it without seeing the graph as well, but it's more about being able to understand and analyze the graph. And then our final question is number 21, that says that many disorders are due to the inability of an individual to break down a particular chemical. Sometimes these disorders can be treated by giving the affected individual the appropriate. So our answer here would be the appropriate enzymes, because we see that the disorders are due to an inability to break down a chemical. And we know that enzymes' entire function are biological catalysts, which can help speeding up chemical reactions. One of the reactions they can do is digestion, or breaking down a chemical. So giving them the necessary enzymes can help them speed up the breakdown of the chemical, helping to solve their issues. Giving them the appropriate chromosome would not be true because everyone is born with all the chromosomes you need. It's not a use of a specific organelle because it's rather systems that break it down, and that enzymes are needed to help break apart the reactions. So it wouldn't be a specific organelle needed. And antigens, which we'll talk about in a later review video, are with the immune system, not breaking down specific chemicals. So our best answer here would be number 1. Okay, and that's all. Thank you for listening.